final round of the 3-minute thesis competition organized by the Young Scientists Association of the National Institute of Fundamental Studies. We'll commence today's proceedings with the National Anthem. Invite the 3MT competition co-chair, Sewanti Kaushalya, to deliver the welcome speech.
Good morning, everyone. Welcome to the 3-minute thesis competition 2021 organized by the Young Scientists Association of the National Institute of Fundamental Studies. I would like to sincerely welcome Dr. Lanka, Dr. Amalka Pinidiyarachi, and Dr. Rajneesh Van Der Khon, our guest judges for today's event. I would also like to welcome Professor Deepal Subhasinha, co-chair of the 40th anniversary commemoration of NIFS, and all other distinguished guests and participants. This year, NIFS is celebrating its 40th anniversary and the 3-minute thesis competition 2021 organized as a part of this celebration. 3-minute thesis competition is organized for the third consecutive year by the Young Scientists Association of NIFS to create a platform to graduate students at NIFS to showcase their research in an informative manner to a non-specialist audience within 3 minutes. I congratulate and wish the best of luck to all finalists who will be competing today. We hope you will have an enjoyable day at 3MT 2021. Thank you.
Thank you, Sewanti. The Acting Director, NIFS, Senior Professor Ranjit Premaralji Silva and the Acting Secretary, Professor Lalicha Sinha, are unable to be present here today due to official matters, but they send their well wishes to all the competitors. Now, Professor Deepal Subasingha, the 40th Anniversary Chairperson, will address the gathering.
Thank you for inviting me to address the gathering. Good morning, everyone. As the co-chair of the 40th Anniversary Committee, I'm delighted to address this gathering and I'd like to welcome you all. I'd like to mention that the Institute of Fundamental Studies was established in 1981 by an act of the Parliament. But it was moved to Kennedy about in mid-80s. In fact, if you had listened to the previous talks by Professor Keith Tennakom, our former director, and the founding director, Professor Chandra Vikramasinghe, you will have known the history of the IFS. Now we call it NIFS. Now, during the last 40 years, we have served the nation in different ways. For example, I have my personal experience with the IFS. When I was an undergraduate, I had my first experience with the IFS when the IFS was organizing an international workshop on astronomy at Taj Summit Hotel, and that was my first experience at the IFS. And also that was my first exposure to the international conferences and community. So that gave me a very remarkable and very unique experience, and developed a big interest in science and research. So I know there are a lot of top class researchers in Sri Lanka who started their careers or who started their taste or they developed the taste for research at the IFS. Because IFS is always a small institute and it always had a small community. So it's like a family. So it's a very good platform to practice, a very good stage to practice the things like public speaking, and also presentation skills and all these things. Then I had an opportunity to join the IFS in late 80s after my graduation. So then I remember after a few years I participated in one of our professional gatherings, and the colleagues and senior people who left the university and joined the other institute, they also presented their findings and things. But I found that the people who presented from the IFS had much better presentation skills, and those days we were using slides and transparent sheets, and they were very good in producing these things and presenting. So that was something I noticed at that time even, because the IFS is giving them an opportunity to practice these things. So that's why I always encourage our young people to participate in this. And also I really appreciate the organizing committee for initiating this and continuing this for the third consecutive year. So please take this as an opportunity to practice and also develop your experience and skills. Without taking much of your time, I will finish my talk, and I wish all the success for the event, and I wish good luck to all the competitors. Thank you.
Now I'd like to invite Dr. Shalini Rajkaruna to give an introduction to today's competition and to introduce the distinguished panel of judges.
Good morning. I'm Shalini Rajkaruna, representing the Young Scientists Association of the NIFS. The 3-minute thesis competition was first introduced to the world by University of Queensland, Australia. Now it is very popular throughout the world, and we are having this competition for the 3rd consecutive year. This year it's very special for us because we are celebrating the 40th anniversary of NIFS, and this event is organized as one of the key events of the series of events we are organizing to celebrate the 40th anniversary. So, on the 14th of December, we had our first round of this competition, and today we have 10 finalists competing for 3 places. And we are so honored to have a distinguished panel of judges, 3 young academics whom we can look up to as young scientists. So, I have the honor of introducing them. On my right hand side, starting from here, we have Dr. Amalka Pinediarachi, Senior Lecturer from the Department of Statistics and Computer Sciences, University of Peradeniya. She graduated from University of Peradeniya with a special degree in Computer Sciences and obtained her PhD in Computerized Image Analysis from the University of Uppsala, Sweden. And next, we have Dr. Rajneesh Van Der Khon, Senior Lecturer in the Department of Biological Sciences, Rajneesh University of Sri Lanka. He graduated with a special degree in zoology from the University of Peradeniya and obtained his PhD in behavioral ecology from Washington University in St. Louis, USA. And next, we have Dr. Tilanka Gunaratna. She is a senior lecturer at the Department of Botany, Faculty of Science, University of Peradeniya. She graduated from the University of Peradeniya with a special degree in botany and obtained her PhD from the University of Aberdeen, UK in ecology. So we are so honored to have you with us today. So let me read out the rules of the 3-minute thesis competition. Presenters are permitted to have only a single static PowerPoint slide. No slide transitions, animations, or movements allowed. No additional electronic media is allowed, example for sound or video clips. No additional props could be used, examples costumes, musical instruments, or laboratory equipment. Presentations are limited to 3 minutes. Presenters who are exceeding 3 minutes will be disqualified. Presentations are to be spoken word, no poems, no raps are allowed. Presentations are to commence from the stage and will be presented on the stage. Presentations are considered to have commenced when a presenter starts their presentation through movement or speech. The final rule is that the decision will be taken by the panel of judges, and that would be final. So, our contestants are ready and the judges are ready, so the stage is yours. Thank you.
Thank you, Dr. Shalini. Before we start the competition, I'd like to kindly request you all to keep your mobile phones on silent. The first presenter is Kulangara Devendra Raja. Kulangara is currently a research assistant in Plant and Environmental Sciences project, reading for her MPhil degree at the National Institute of Fundamental Studies. She obtained her BSc special degree in Environmental Conservation and Management from University of Kelaniya and joined NIFS in 2021. Her research focus is on nutrition management, environmental remediation, and phosphate biofertilizers. Kulangara will be presenting today on her topic, Are You Carbon Neutral?
Good morning. Are you neutral? The specific question is, are you carbon neutral? I am able to tell one true fact about all of you sitting here. Don't worry, I am not a mind reader. The fact is, you all today morning travel here using either a vehicle or on foot. Am I right? While you are traveling, have you noticed the street trees in the both sides of the road? Have you ever think why those trees are there? Is it for beauty? Is it for shade? Or is it for soil erosion? I took my steps out of your shoes, and this is where my research comes into the picture. It is on evaluating the carbon storage potential of selected street trees in the urban city. When we consider Sri Lanka as a whole, the role of carbon storage in these trees are still remain uncertain due to the absence of proper selection criteria. So what is the need? We need a proper selection criteria. How to overcome that? By replacing it with a better species. So my research, I focus on carbon storage as a selection criteria. I analyze the storage of me, Ahala and Karanda, which are the most abundant street trees in the urban cities. I found that me has the highest carbon storage capacity. So I recommend Sri Lanka to plant more me trees in the streets in order to neutralize your carbon emission. More than that, I have derived and validate the equation to calculate the carbon storage. If you know the diameter of the tree, you will be able to calculate the carbon storage. So now tell me, is these trees are only for shade, only for beauty, only for soil erosion? It's more than that, right? It is also for carbon storage. So now think with me. I am able to tell your carbon emission. Go and hug a tree, you know the diameter of the tree. Now you have my research idea, you will be able to calculate the carbon storage. You know the storage, you know the emission. Now tell me, are you neutral? Still you have time. If not now, when? If not you, who? Thank you. Over to you.
Our next competitor is Maheshika Pereira. Maheshika Pereira is a PhD research assistant with the Plant Stress Biology and Molecular Genetics project at the NIFS. She obtained her BSc equivalent special degree from Institute of Chemistry Ceylon in 2014. She began her career with Sri Lanka Institute of Nanotechnology as a research assistant. She joined NIFS in June 2019. Maheshika's research focuses on precision nitrogen management through environmental benign hybrid nanomaterial. Her research interests include nanotechnology, plant nutrients and plant physiology, and functional genomics. Her topic for today is feeding crops as nitrogen feed.
Fertilizers. As Sri Lanka, we all are affected by this fertilizer issue these days. When I mention the word fertilizers, I guess the first word come to your mind is urea. So what is urea? Isn't there any proper nitrogen fertilizer system? Let's talk about this. Plants and animals, we both are living organisms. As human beings, we are growing up with food. But if I go back to our childhood, do you remember how we started our meal consumption? Did we eat whole plate of rice from the day one? No. After our mother's milk, we have started our meal consumption by eating that rice bulk, but very little amount. This means when we are growing up, we need foods in different textures and in different amounts. Therefore, how about plants? They are also like us. They also have different nutrient uptake patterns when they are growing up. Therefore, we need to feed them accordingly. That's where my research comes in. As urea is well known as common nitrogen fertilizer, could plant uptake this urea as it is? The answer is no. Plants could not. With the help of soil microorganisms, this urea converts into nitrate and ammonium. Those are the only two forms of nitrogen that plants can take up. By having that knowledge, I found out the exact ratios of nitrate and ammonium which can be taken up by the rice plant at different ages. And also, with the help of nanotechnology, I designed and developed a nitrogen fertilizer system. The beauty behind this fertilizer system is it can release nitrogen only upon the plant requirement. Not less, not much, just to the need. I'm pretty sure plants would love to have the exact level of nitrogen when they are growing up. If so, happy plants will give more yield and will make farmers happy. And the end of the day, that will the environment, the balanced nitrogen environment will make the environment happy too. Thank you.
Our next presenter is Medhavi Abesinghe. Medhavi is currently an MPhil research assistant in the Earth Resources and Renewable Energy Project at NIFS. She received her BSc Special Degree in Geology from the University of Peradeniya in 2018 and joined NIFS in 2020. Her research focuses on geophysical and hydrogeological studies along with structural geology in geothermal springs of Sri Lanka. Her research interest includes exploration geophysics, metamorphic petrology, structural geology, geochemistry, GIS, and remote sensing. Today, Medhavi will be presenting on modeling of subsurface at Kapurala geothermal spring.
In Sri Lanka, there are several hot water springs located in the eastern and southeastern part of the country. Some of them have developed for tourist attraction and some for villagers' use. But this is like a gold mine to Sri Lanka because we can utilize this heat or geothermal energy for better uses such as generating electricity. Up to date, we have wind, thermal and hydropower plants. But when compared with them, generating electricity using this method could be more advantageous because it is more environmental friendly, has a great efficiency, and also geothermal power plants have a long life, more than 50 years. Except for generating electricity, we can utilize this heat to dry agricultural crops, for sugar and paper industries, and also for warm pools and spas. And for all of these, we have to know from where this water comes from and how this water gets heated. Water can get heated from several ways. When we are going deeper into the earth, usually temperature increases. So, when the water returns to the surface from the deep earth, they can be comparatively warmer than the surface water. And also, water can get heated from volcanic activities. Except to these, there can be other reasons too. Therefore, my study focuses on finding the heat source, location, expansion of the reservoir beneath the ground, and also the flow paths which connect the reservoir to the surface. For that, I am using Kapurala hot water spring located at Ampara district because it has recorded the highest temperature so far in Sri Lanka. I am using resistivity and magnetic properties of the soil and rocks of the hot spring, of the ground of the hot spring, to study the structure related to the reservoir. From that, I am going to make a model of the reservoir and the surroundings. From the model, we can assume the geothermal potential of the reservoir. And if the outcomes are good, we can develop a model to construct a power plant. Thank you.
Our next contestant is Harshini Nandishwamy. Harshni Nadishami joined as a PhD research assistant to the Nutritional Biochemistry project at the NIFS in 2021. She received her BSc Honours degree in Food Science and Technology from the University of Perdini, Sri Lanka in 2015 and an MPhil degree in Food Science and Technology from the same university in 2018. Her research interests lie in the area of nutritional biochemistry and functional and nutritional properties of food. For her PhD, she will be focusing on nutritional status and prevalence of early childhood caries among Sri Lankan children. Her topic for today is nutritional value of fruits and vegetables grown in Sri Lanka.
How is your preference to eat fruits and vegetables? Please hands up if you eat 3 to 5 vegetable portions and 2 to 3 fruit portions per day. See how many of you acquire your meals with no or very few servings of fruits and vegetables. Among Sri Lankans, the average intake of fruits and vegetables is as low as 0.43 and 1.73 portions per day respectively. Above all, only 3.5% of adults consume recommended portions of fruits and vegetables. However, fruits and vegetables play a major role in human nutrition as they are rich sources of essential nutrients and bioactive components. They are attributable for disease prevention, health promotion, providing you a healthy body image. But we do not take enough effort to include these fruits and vegetables into our diet. As a consequence of this unhealthy consumption pattern, people are progressively subjected to chronic diseases such as diabetes, cancers, high cholesterol, heart diseases, obesity, etc. The problem is very serious as 71% of total deaths in Sri Lanka is due to these chronic diseases. In addition to that, Sri Lankan food composition database of fruits and vegetables, which was used by national and international organizations such as FAO and WHO for nutritional claiming, nutritional surveys, developing policies and programs, was outdated, and it was designed in 1979 that highlighted the timely requirement of developing updated and precise food composition database of fruits and vegetables in Sri Lanka. Furthermore, though Sri Lanka is inherited with wide variety of fruits and vegetables, people are not aware what fruits and vegetables are highly nutritious. Therefore, my MPhil research was designed to investigate the nutritional value of fruits and vegetables grown in Sri Lanka. For that, different selected fruits and vegetables were collected from all over the country and prepared into dried and powdered samples. Those samples were analyzed for carbohydrate, protein, fat, dietary fiber, vitamins, minerals, and fatty acids. According to the results, Udapal and Sabosop among fruits, Tibatu and spine gourd among vegetables, Thampala and Kathurumurunga among leafy vegetables were nutritionally superior. As another outcome of this study, now we have updated food composition database, which was a long felt need and a request from international organizations such as FAO and WHO. Thank you very much for your time and your attention.
Our next competitor is Jayani Kalinga. Jayani is currently a research assistant in Natural Products Project, reading for her MPhil degree. She obtained her BSc Special Degree in Botany from Southeastern University of Sri Lanka and joined NIFS in 2020. Her research focuses on identifying and isolating bioactive secondary metabolites from endophytic fungi in medicinally important Sri Lankan flora. Her research interests include plant biochemistry, natural products chemistry, and fungal biology. Today, Jayani will be presenting on her topic, Analysis of Secondary Metabolites in 5 Endemic Medicinal Plants in Sri Lanka.
Mr. Tree behind me is asking a question. Did you know that plants produce beneficial chemicals for humans? If not, think about Ayurvedic medicine. It completely relies on the medicinal plants of a region. So unknowingly, we have been taking the advantage of phytochemicals for many generations. Phytochemicals are two types, primary metabolites and secondary metabolites. Primary metabolites are very much needed by the plants for their own growth and development. Secondary metabolites are not essential for a plant. Surprisingly, most of these secondary metabolites have a physiological effect on humans, which means some can reduce the blood sugar level, some can enhance our memory, whereas some can slow down the aging process. Secondary metabolites with such amazing properties are known as bioactive compounds, and there are 4 classes of them: phenols, flavonoids, alkaloids and tannins. But how do we know which medicinal plant is having which of these chemicals? That is where my research came in. I was focused to determine the quantity of secondary metabolites, or these amazing chemicals, in 4 endemic medicinal plants of our country, namely Hembovitia, Idda, Irivaria and Daruharindra. I took the extracts from dried parts of these plants and performed a series of chemical experiments. There, I found that Hembovitia is having a significant quantity of phenols and tannins. Daruharindra is rich with alkaloids and Irivaria is rich with flavonoids. So based on my results, these plants should be further studied to isolate possible compounds, determine their bioactivity and to illustrate their structures so that they can be used to manufacture a novel drug in the future. What important in that is the drugs produced from naturally occurring compounds are known to have more effect on the disease and less side effects and less costly. If we are able to do that, we can bring lots of money to our country because they only grow in here. By saying that, we should also keep in our mind to conserve these for future generations, because based on my research, these endemics can be called as natural reservoirs of bioactive compounds. Thank you very much.
Our next contestant is Tilakshi Paranvithana. Tilakshi Paranvithana is currently attached to the Microbiology and Soil Ecosystem Research Project at NIFS as an MPhil Research Assistant. She received her BSc from the Subsurface University of Sri Lanka in 2018 and joined NIFS in the same year. Her study at NIFS focuses on development of baseline soil information system for soil carbon and other nutrients for paddy growing soils in Sri Lanka. Her research interests include environmental sciences, soil chemistry, data modeling and spatial analysis. Her topic for today is healthy soil, wealthy nation.
Well, first of all, if someone asks from you what would be the most prominent environmental disaster at the present, definitely the first thing that comes into your mind is the global warming, because almost all other environmental disasters are closely associated with this problem. Mainly, excess carbon dioxide gases in the atmosphere is the major contributor for this problem. Therefore, with the recognition of the soil as a valuable reservoir that can store the excess carbon dioxide in the atmosphere, the worldwide researchers started their studies on this below ground soil carbon content. On the other hand, with the rapidly growing population, to fulfill their food demand, people tend to apply vast amount of different fertilizers to their cultivation. But unfortunately, they don't have enough knowledge to understand what is the fate of these fertilizers within the soil without proper organic matter management. That's where my research began. The research mainly focused on to develop a soil carbon map that showing the soil carbon contents and its distribution across the paddy growing soils of Sri Lanka. For this purpose, soil samples are collected as represent whole Sri Lanka
and their soil carbon content and other chemical physical properties were analyzed. By using this analyzed data with the different climatic, topographic, soil type information and the vegetation index data, a soil carbon model was developed. By using this model, soil carbon contents across the Sri Lanka was predicted. Further, this will be the first-hand information that need to feed the National Carbon Accounting System in the Sri Lanka. Also, this will be a good initiative to establish soil-based carbon buying and selling concept with other countries. Further, the identified significant relationship with soil carbon with other nutrients will be used to develop another kind of model that can be used for the site-specific fertilizer recommendations, which is perfectly matched for the local soil conditions. This will in turn help to reduce the unnecessary expenses on these fertilizers. Therefore, you can understand, really a healthy soil is a wealth for a nation. So, let's stand together to save our planet.
Our next presenter is Upesha Sirivardhana. Upesha is currently an MPhil research assistant with the Natural Products project at NIFS. She received her BSc Honours in Chemistry from University of Sri Jayawardenepura in 2019 and joined NIFS in 2020. Her research focuses on the study of antifungal, enzyme inhibitors and other bioactive compounds from selected medicinal plants in Sri Lanka. Her research interests include organic chemistry, spectrometry and natural products chemistry. Today, Upesha will be presenting on her topic, Coir red liquor treatment by Fenton process.
As Sri Lankans, coconut husk fiber or coir fiber is not a strange word for us. Basically, we use coir fiber in most of the products we use in day-to-day life. But have you ever thought production of coir fiber causes severe water pollution? Well, to produce coir fiber, coconut husks are immersed in water and the organic compounds present in the husks are leached into the surrounding environment. This water is called red liquor. Now, red liquor has a very offensive odor, color, and cause very bad effect on the environment. Therefore, it is crucial to treat red liquor before discharging into the environment. But for an industry, waste management does not create any profit. Therefore, the treatment method should be really cost effective. This is where my research comes in. In my research, we looked at a method to treat red liquor using simple equipment, chemicals, and mild operating conditions. Fenton process is a simple reaction between ferrous and hydrogen peroxide, which generates hydroxyl radicals. Hydroxyl radical, very reactive oxygen species. We measured the pollutant level of the sample using COD value, chemical oxygen demand. Now, the pollutant level of the sample is high. The COD value is high. Usually, the water we drink has a COD value around 0. Now, red liquor had an initial COD value around 2000 ppm. We treated red liquor using different ratios of ferrous and hydrogen peroxide. Now, the results we obtained were really exciting. From only 1 milliliter of 10% hydrogen peroxide and 1 milliliter of 0.1 m ferrous, we could achieve over 90% COD reduction, significant odor removal and color removal within 5 hours. Which means now we can safely discharge this water into the environment. So, I think with the help of my research, we will be able to preserve one of our planet's most valuable resources. Thank you.
The next contestant is Sumana Sasivan. Sumana is an MPhil research student in the Molecular Microbiology and Human Disease Research Group at NIFS. She received her BSc Honours in Medical Laboratory Sciences from University of Jaffna in 2015 and joined NIFS in 2020. Her research focuses on identification of urinary biomarkers in diabetic and hypertensive chronic kidney disease patients in Sri Lanka. Her topic for today is early detection of kidney failure using advanced molecular techniques.
Kidney failure. Let us think a few seconds why we all are worrying about kidney failure. Shall we live without kidney? If anyone asks this question to me, I say no, because kidney is the only organ in our body which remove all the waste from the body. There are a lot of factors which damage the kidney. Among those, two factors which severely damage the kidneys are number 1, diabetes, number 2, high blood pressure. So, 6 out of 10 people in Sri Lanka having diabetes. So first we have to think how to control these factors. Are you ready to control your sugar level? No, you can't. Even for me, I also can't, because we all are living with a sedentary lifestyles. So what is the next options? We have to think of treatment. So only 2 treatment facilities are available to treat the kidney disease. One is dialysis, that means we just remove the blood from the patient body and again clean it and again inject into the patient body. Second option is transplantation, that means we replace the kidney with a new kidney. So, however, these techniques are not available to everyone at all, since we are living with limited resources. So, see how many people are dying without reaching the treatment facilities in Sri Lanka. So, in this point, the journey of my research was started. So, I mainly focused in my research to detect the kidney disease in patients who have diabetes as well as hypertension as much as early, because the existing methods are not available to detect the disease early. So what I did in my research is, I collected urine samples from the CKD patient, kidney disease patient, and isolated the genetic material from that. And using a highly advanced molecular techniques, I compared that genetic material from the healthy individuals. Interestingly, I found that a single gene that responsible for the kidney damage that could be used to detect the kidney disease in early, especially those who have diabetes and hypertension. Finally, my research findings would help to reduce the number of deaths occurred because of kidney failure in our country. Thank you.
Our next presenter is Savani Ulpathakumbura. Savani is an MPhil research assistant in the food chemistry project at NIFS. She received her BSc special degree in food science and technology from University of Peradeniya in 2020 and joined NIFS in the same year. Her research focuses on anti-hyperglycemic, anti-obesity and antioxidative properties of selected edible leafy plants of Sri Lanka. Her research interests include food chemistry and food biochemistry. Today, Savani will be presenting on the topic Quality Analysis of Turmeric from Sri Lanka and Myanmar.
Let's do a simple imagination. Can you all just think about your favorite cuisines like coconut milk curry and your biryani and your favorite pancake, but not with that nice yellow color? What would happen to your appetite? Not good. Is it correct? So yes, life is way better with turmeric. Turmeric tightly attached to our cuisine styles as a spice, colorant, as well as a preservative agent. Have you all aware of curcumin?
curcumin is the principal component present in turmeric, which is responsible for its biological properties, including anti-diabetic, antioxidant, anti-cancer, anti-inflammatory, and many other biological properties. Locally, very high demand exists for turmeric for local cuisines. However, as we all know, adequate production within the country is not ensured. Therefore, a request has made by food industry to grant permission to import turmeric from Myanmar. That's where my research comes to monitor the situation at the very first place to take the decision on importation. In my study, I compared the quality parameters of turmeric imported from Myanmar with locally grown turmeric. So, especially for the curcumin content as well as for the other quality parameters, it was really amazing. The results clearly showed that there's a considerable quality differences among both turmeric from Myanmar as well as from locally grown, especially for the curcumin content as well as for the other quality parameters. According to the internationally recognized specification, turmeric should contain at least 3% of curcumin. Interestingly, our turmeric contained curcumin range from 3.9 to 6.9%, while it was only 1.3% for turmeric from Myanmar. Even it is not requirement of half of the amount. So, based on the results, there is a problem with the quality of the turmeric from Myanmar. So, in order to fulfill the local demand, it is better to go for extensive cultivation within the country, since we clearly notice that our turmeric is good in quality. Thank you.
Now we have come to the end of the competition. We'd like to invite you all for tea at the cafeteria, and we shall meet back at 11:05 for the awarding ceremony.
Thank you so much for your patience. So now the judges have finally come to a decision. Before we get to know about the decision, I would like to invite the judges to come on stage and give their general remarks on the presentations of our research assistants today. And first I would like to invite Dr. Amarka Pilidiyarachi on stage.
Hello everyone. First of all, let me start by thanking Shalini and IFS for inviting me to be part of this wonderful occasion. It was a great experience for me. And I must say, coming to IFS is like coming to an old home. My first visit to IFS was already in 1991, when they had the school science program. So even though Shalini says we are young, of course we are young at heart, but we can guess our age. So it's actually a very pleasant experience to come to IFS, as always. Talking about today, let me start by congratulating all the presenters for a job well done. I was actually very impressed to listen to your presentations. They all were equally good. And that made our life very much difficult because we had a very tough time giving marks. So you can take my word for it that you all have done an excellent job, irrespective of who become the winner today. So let me congratulate all of you on that. And I was amazed to see how well you presented, and you had no stage fright at all. And you were right on time, and your time management was very good. Nobody exceeded the 3 minutes given. And you started very nicely, and you conveyed your messages very nicely, and you ended the presentation very nicely. So all that was very good plus points for all of you, and I was very happy to see that. Few comments for improvement, because as always, any great presenter can improve themselves. So one thing we noted was that I think all the other judges also would agree with me. Your slides could have been improved a bit more, because the whole idea of this 3-minute competition is to give a popular science presentation of your research. So you have always keep that in mind, but having said that, you should not also dilute your content of the presentation too much as well. It should still convey the message, but it should be easily understood by the audience as well. Now, we like some of your staff said, we all are still coming from science backgrounds, but the whole idea is even if somebody from out there on the road comes and listens to your presentation, they should still be able to understand what you are saying and what you have done in your research and what usefulness it has to the general public out there. So that idea, you probably have to think a little bit more when you are preparing the slides. And the presentations themselves, when you speak, like I said, you did a great job. But sometimes you tend to spend a little too long time on like giving the background, but not so much on the exact research. Of course, we still have to give it in a popular science manner, but still, I would have preferred if you explain in easy to understand manner what you have actually done as the research work. So a little bit more on that probably would have been better. That's my personal idea. But all in all, I have enjoyed your presentations thoroughly, and I would like to wish you all the best for your future presentations as well, and congratulate you once more. Thank you.
Thank you, Dr. Amalkan, and I would like to invite Dr. Rajneesh Van Der Khon on stage.
Hello. First of all, I'd like to thank the IFS for having invited me to this particular event because I really enjoy, whenever somebody sends me an invitation to attend something in another university, I always take it up because I see it as an opportunity to go and learn something, to see what other students are doing, what others are trying to achieve, because it enriches our system as a whole. So we all have as researchers and educators, we all have similar goals. We want to mentor students, bring out the best in them, and I think the NIFS has succeeded in doing that. So when I met Shalini this morning, I just after the presentations, I asked her, is this tied to their jobs? I mean, will they get fired if they refuse to do a 3-minute presentation? So then she said, no, this is, they volunteer to do this. So, irrespective of whether, you come first, second or third, I would like to congratulate all the participants on their bravery. Because if I was given the opportunity to opt out of this, I would have done it without batting eyelid. Because sometimes summarizing your work in a way that's understood by the public in a very short period of time is a very daunting task. It's not easy. So in that respect, I really like to congratulate you all, and you're a brave lot. As with anything we do, there's always room for improvement. So I think Dr. Amalka has already highlighted some of these issues. I mean, these are things that I'm pretty sure you all are even aware of. But in addition to what she said, some minor issues that I saw was there was some in certain presentations there was a disconnect between the title and what was being presented, a very slight one. Other than that, I found your presentations to be of really good standing, and I really congratulate you and your mentors for helping you all achieve this level of standard. So once again, congratulations, and I wish you all the very best. I hope you all remain in science, right? And for the sake of this country. And I congratulate the NIFS for organizing an event of this kind. Thank you.
Thank you, sir. Now I would like to invite Dr. Lanka on stage to give her remarks.
Good morning to all of you. I hope yes, it's a very pleasant morning for all of us, and thank you, Shalini, and the organizing committee of this event in IFS for giving us this opportunity. Actually, this is the first time I participated for such an event, and I found it really thrilling to see the next generation of researchers. Their actually communication skills and the stage presence. when we were at your age, if we were given the task, we would have been terrified. But you all took up the challenge, and with the help of your supervisors, I'm sure you did a marvelous job. As the other two judges mentioned, they mentioned a lot of remarks, room for improvement as well. I congratulate all of you actually. It was a really difficult task for us to give the marks, because all of you did a great job. All had good eye contact, good stage presence. Some of you were really confident on stage. And so researchers, next generation of researchers, this is a time for peer learning as well. We all learn, we all are lifelong learners, we learn from you as well. So learn from other researchers. It is not only doing science that is important. At my age, I feel we have to connect with the society, what we find. In the 10 presentations, your speeches you did today, I found really important science that you could easily write up to newspapers, local newspapers. I urge you all, along with your supervisors, please write. We have been, as university academics, trying to popularize this culture, to disseminate knowledge to the society, because they would love to know what are the best vegetables to eat with high nutrients. What are the things we can do to identify kidney diseases before we have to go to the doctor. And so it's high time. I hope you all will work on it, and you will lead the way, and we will also try in university level to disseminate knowledge. So with the scientific community only we can lead Sri Lanka as scientists. If not, our voice won't be heard. Saying that, there were a few remarks I want to comment on. Room for improvement. As we all know, Champions Breakfast is feedback. So we need feedback. And one of the main comments I have for all of you, most of the slides, it's for the PowerPoint slides, they were too simple, as one of the judges mentioned, diluting your research. So try to work on slides. One plant, although I am a botanist, I was keen to know what is that Devadar or something like that. What is this plant? And there was no photograph. So the best plants, maybe that one could have been improved. Not only that, all the slides I thought could have improved a little bit more to give more information. Okay. With that, I congratulate all the next generation of researchers, and I'm really happy that IFS has taken this challenge to organize 3-minute thesis presentations. Thank you for giving me this opportunity.
Thank you, Madam. Thank you very much for taking up the challenge to judge our students. And now the moment of truth has come. I have the final results in my hand. So as all of you could see, it was a very difficult task for the judges to select 3 winners. So as I mentioned in the beginning, the judge's decision is the final decision according to the rules, international rules of 3-minute thesis competition. So we have a winner, and also we have 2 second places. So the second and third places will be divided among the 2 second place winners, and the second runner-ups, which means the third place winners, will be given 2 merit awards. Every first place is given a cash prize of 10,000, and as we have 2 second place winners for the second place, we have divided the cash prize between 2. So the each winner for the second place will get a cash prize of 5,500 rupees. Apart from that, the most important thing is that the winner will get a gold medal, and the first runners-up
will get 2 silver medals. For the moment, we are going to share the silver medal among the 2 second runners-up and the first runners-up, and the 2 second runners-up will be awarded certificates of merit. I hope it was clear. So let me announce the winners. First, I'm going to announce the names of the second runners-up who will be awarded merit certificates. And I would like to invite Professor Deepa Subasinghe, the co-chair of the 40th Anniversary Commemoration Committee, on stage to give away the certificates and awards. Second runner-up for the Merit Award is Upesha Sirivardhana. Merit Award. Certificate. The next second runner-up for the Merit Award is Savani Ulpat Kumura. Now, I'm going to announce the first runners-up who are going to share the second place and the silver medal and cash prize of 5,500 each. This was a very difficult decision to make for the judges. So let me announce the second place. T. Kulangara. Kulangara will be sharing her silver medal with the other second place winner, so they will sanitize the medal and give away to the second. Okay, let me announce the next first runner-up who will be sharing the silver medal and a cash prize of 5,500 rupees. The next winner is Harshani Nadishani. So it's the time to announce the name of the winner for the gold medal, the first prize and a cash prize of 10,000 rupees, who did the best today. So let me announce the name of the first place, the winner. The winner of the 3-minute thesis competition of NIFS for the year 2021 is Jayani Kalinga. Thank you very much, Professor Depal. I congratulate all the winners, and it is time to say thank you to the judges who did a marvelous job. And I would like to invite Professor Lakshman Disanayak, representing the Board of Governance of NIFS, on stage to give away the tokens of appreciation to the judges. First, I would like to invite Dr. Amalka Pinidya on stage. Let us give a big round of applause to the judges. Next, let me invite Dr. Rajneesh Van Der Khon. Then I would like to cordially invite Dr. Lanka on stage. We have some certificates to be given to the finalists and the participants. I would like to invite Professor Deepa Subasinghe again on stage to give away the certificates. Certificates for the finalists who presented their 3-minute thesis presentation today. Maheshika Pereira, Medhavi Abesinghe, Tilakshi Paranamithana, Sumana Sasivan. We have another 4 certificates to be given to the participants. Nirasha Atapattu, Lasanga Amarasena, Hiran Kankanange, and Mayumi Tilakaratna. Thank you very much, sir. While congratulating all the winners and the participants, I would like to invite the co-chair of the Young Scientists Association, Ms. Maheshika Pereira, to deliver the vote of thanks.
Ladies and gentlemen, it gives me an immense pleasure to deliver the vote of thanks of this very important day on behalf of the Young Scientists Association of National Institute of Fundamental Studies. Firstly, I would like to express our sincere thanks to the Chairman of NIFS, Professor Atula Sumathipala, the Acting Director of NIFS, Professor Ranjit Premaladi Silva, and also the Acting Secretary of NIFS, Professor Lalit Jayasinghe, for their encouragement and guidance. And also, I would like to express our gratitude to Professor Deepa Subasinghe and the committee of the NIFS 40th anniversary celebration for their immense support in organizing this event. A special mention goes to the Science Education and Dissemination Unit of the NIFS for their dedication and the enormous cooperation in making this event a reality. We would also like to express our heartiest appreciation to the judges of the preliminary round Professor Rohan Veersurya, Dr. Kumari Tilakaratna, and Dr. Ruvini Liyanage of the National Institute of Fundamental Studies, and the judges of today's finals, Dr. A.M.T.A. Gunaratna, Dr. Amalka Pindyarachi from University of Peradeniya, and Dr. Rajneesh Van Der Khon of Rajarata University, Sri Lanka. It was an honor and privilege to have you as judges of this event. And also, I would like to thank our former director, Professor Saman Senavira, to introduce and encourage us to do this 3-minute thesis competition at NIFS. Also, I would like to thank Dr. Shalini Rajakaruna, Dr. Lakma Jayaratna, the advisors of the YSA NIFS. Organizing this event would not have been possible if not the dedication and the hard work of the members of the YSA of NIFS, and I would like to thank the organizing committee of the 3-minute competition of the YSA to their untiring effort. We would like to thank all those who have participated in this competition, and we congratulate the winners. Finally, we sincerely acknowledge the University of Queensland for authorizing YSA of NIFS to conduct this 3MT competition. I wish all a pleasant day. Thank you very much.
With this, we conclude today's event, and we hope to see you next year with interesting presentations like this. Thank you very much for your participation.